All right, today we're going to dive into one of the absolute cornerstones of genetics, Mendel's law of independent assortment. Now, I know it sounds a little intimidating, but it was all figured out through a really clever experiment with pea plants, and it helps us understand how we inherit our traits. So, think about it for a second. Do your traits come in a package deal? Like, if you get your mom's hair color, does that mean you automatically get her eye color too? Or is it more like a buffet where you can pick and choose traits independently? That's exactly the puzzle that the great Gregor Mendel wanted to solve. So here's our game plan. We'll start with the puzzle Mendel was facing, then we'll walk through his experiment step by step. We're going to follow the results through 2 whole generations of pea plants, which will lead us right to this core idea of independent assortment and the famous 9:3:1 ratio that proved it all. See, Mendel was already on a roll. He'd figured out the basics of how single traits, like just flower color, were passed down. So the next big question was, what happens when you're tracking 2 different traits at the same time? Does one affect the other? To figure this out, he decided to look at 2 really clear traits in his pea plants. The shape of the seed, was it round or was it wrinkled? And the color of the seed, yellow or green. The million-dollar question was, are these traits linked together, or do they get shuffled around independently? Now, every good experiment needs a really clean starting point. For Mendel, that meant using what's called the parental, or P generation. And these weren't just any old plants. He needed plants that were true breeding, which is a fancy way of saying that for generations they only ever produced offspring exactly like themselves. And here are our 2 star players. On one side, we have a plant that's true breeding for round yellow seeds. That means if it pollinates itself, it only makes more round yellow seeds. He crossed that one with another true breeding plant that only ever made wrinkled green seeds. These 2 are our starting line. Okay, to keep track of all this scientifically, we use symbols for the different versions of a gene, which we call alleles. For seed shape, we use a capital R for the dominant round allele and a lowercase r for the recessive wrinkled one. Same deal for color, capital Y for dominant yellow and lowercase y for recessive green. A dominant allele basically just overpowers the recessive one when they're paired up. And this brings us to a super important idea, the difference between genotype and phenotype. The genotype is the actual genetic code, the letters like RRYY. The phenotype is the physical trait that you can actually see. So for our parent plant, the genotype is RRYY, but its phenotype is round and yellow seeds. So Mendel plays matchmaker. He takes the pollen from one plant and uses it to fertilize the other. What do you think the very first generation of offspring, the F1 generation, is going to look like? And check this out. The result was incredible. Every single one of the F1 plants produced seeds that were round and yellow. Even though their genotype, their genetic code, was a hybrid RRYY, it had alleles from both parents, their phenotype only showed the dominant traits. The wrinkled and green traits seemed to have just vanished. But this is where the real magic happens. That first generation looked all the same, but what was hiding inside those hybrid genes? To find out, Mendel let these F1 plants self-pollinate. He basically crossed an RRYY plant with another RRYY plant. And the results from this second generation, the F2, were the key to the whole puzzle. Now, before we can predict what those F2 plants will look like, we need to understand how these F1 hybrids create their sex cells, or gametes. How the different alleles get sorted into these cells is the absolute core of this whole thing. So during a process called meiosis, when that RRYY plant is making its gametes, the 2 alleles for shape, the big R and the little r, get separated. Half the gametes get the R and the other half get the R. And the exact same 50-50 split happens for the color alleles. Half get the big Y and half get the little y. And here is the absolute game changer. The way the shape alleles R and R get sorted is completely independent of how the color alleles Y and Y get sorted. So a gamete that gets a big R has an equal chance of getting a big Y or a little y. This gives us 4 equally possible combinations for the gametes: RY, 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 and RY. Because this sorting process is totally random, each one of these 4 gamete types gets produced in equal numbers. There's a 25% chance for any gamete to be RY, a 25% chance to be RY, and so on for the other 2. This equal probability is the foundation for everything that comes next. Okay, so we've got 4 kinds of male gametes and 4 kinds of female gametes, all happening with equal frequency. How in the world do we figure out all the possible combinations for the offspring? Well, for that, we use an amazing little tool called a Punnett square. And here is the whole shebang laid out for us. You can see we list the 4 possible male gametes across the top and the 4 possible female gametes down the side. Each one of these 16 boxes shows us a possible genetic outcome for an offspring. So for example, if an RY sperm meets an RY egg, boom, you get the RRYY genotype in that top left corner. Or if an RY from the male fertilizes an RY from the female, you get RRYY. All right, so let's go through these 16 boxes and just count what we can see, the phenotypes. Remember, as long as a plant has at least one big R and one big Y in its genotype, its seeds will be round and yellow. So when you count them all up, you find something amazing. 9 are round and yellow, 3 are round and green, 3 are wrinkled and yellow, and just 1 is wrinkled and green. And that gives us the famous dihybrid phenotypic ratio, 9 to 3 to 3 to 1. This isn't just a coincidence. It's the mathematical signature that proves 2 different genes are being inherited independently of each other. It's really beautiful when you think about it. Okay, now this is the really cool part. If you just look at seed shape in that big square, you'll find 12 round ones and 4 wrinkled ones, which simplifies to a 3-to-1 ratio. And if you just look at color, you find 12 yellow and 4 green, also a 3-to-1 ratio. So that big 9-3-3-1 ratio is literally just those 2 simple independent 3-to-1 ratios multiplied together. That's the mathematical proof right there. So, from this one beautifully designed experiment and its incredibly clear mathematical results, Mendel was able to come up with his second major law of inheritance. And there it is, the law of independent assortment. All it really means is that the alleles for seed shape get sorted into gametes totally randomly without caring one bit about which alleles for seed color are getting sorted. They are not a package deal. So let's do a super quick recap. The signature result is that 9-3-3-1 ratio, and that comes from the 4 equally likely gamete types, which combine to give us 9 unique genotypes and 4 unique phenotypes in that second generation. All these numbers are the direct result of independent assortment. So we've just laid out a fundamental law of genetics. But you know how science works, right? For almost every rule, there's an interesting exception. Mendel's law is perfect for genes on different chromosomes, but what happens when 2 genes are physically located right next to each other on the same chromosome? Well, that's a story for another time. Thanks for joining this explainer.